Good afternoon, dear students. I am Lims Mary Joy, Associate Professor from Department of Pharmacognosy in Nirmala College of Pharmacy. Today we will discuss on the topic carbohydrates. When we think about the carbohydrates, we are taking the carbohydrates in our daily life, like the rice, then wheat, maize, etc. So we are coming across various type of carbohydrates in our daily life. Now let us see what are carbohydrates. When we define the carbohydrates, previously the carbohydrates were defined as those compounds which contains carbon, hydrogen and oxygen. But later it was proved not true in all cases. Like for example, if you take the formaldehyde, HCHO, it also contains the carbon, hydrogen and oxygen, but they are not carbohydrates. So later they modified the definition of carbohydrates and the carbohydrates are polyhydroxy, aldehydes and ketones. Now we move on to the classification of carbohydrates. How can we classify these carbohydrates? Carbohydrates are classified into three different categories. First one, monosaccharides, oligosaccharides and polysaccharides. Monosaccharides, as the name says, it contains only one sugar moiety, and this is known as the simple sugars. And these monosaccharides can again be divided into different types based upon the number of carbon atom and based upon the functional group present. And based upon the number of carbon atom, if it contains two carbon atoms in the single sugar moiety, it is known as the biosis. And if it contains three carbon atom in the particular monosaccharide unit, it is known as triose. And if four carbon atoms are present, tetrose, five carbon atoms, pentose, and so on. Similarly, based upon the type of functional group, they are divided into two types. If it contains an aldehyde group, it is known as the aldose. And if it contains the ketone group, it is known as the ketosis. And the next category of the classification of carbohydrates is oligosaccharides. That means if the sugar or the carbohydrate contains 2 to 10 sugars, it is known as the oligosaccharides. And these oligosaccharides are again classified into different types based upon the number of sugars. So if the carbohydrate contains two simple sugar moieties, it is known as the disaccharides. And a very good example is the sucrose, which is found in the sugar cane. It is composed of two simple sugar moieties, that is the glucose and fructose. Similarly, in the milk, we can see the lactose, which is again made up of glucose and galactose. And the malt sugar is known as the maltose. It is made up of two simple sugars, that is the glucose. So here, two glucose units are combined together to form the maltose. Then next one is the tetrasaccharides. So as the name says, tetra means four. So tetrasaccharides are composed of four sugar moieties. An example is statiose, which is obtained from manna. Then trisaccharides, again tri means three. So it is composed of three sugar moieties. Very good example is raffinose, gentianose, etc. And the next category coming under the classification of carbohydrates are polysaccharides. So polysaccharides, they contain indefinite number of sugar moieties. And very good examples are starch, inulin, cellulose, xylan, etc. And this is again broadly divided into two types, that is homopolysaccharides and heteropolysaccharides. Homopolysaccharides are completely made up of the similar type of sugar moieties, whereas the heteropolysaccharides, they are made up of different type of sugar moiety. For example, it is composed of glucose and fructose. Whereas in case of homopolysaccharide, it is made up of one carbohydrate is made up of only glucose moieties, then it is known as the homopolysaccharide. Then we'll move on to the important carbohydrates of plant origin. Under this, first we'll see the agar, which is a carbohydrate which is obtained from the seaweed, which is known as the Gelidium amansi, and belongs to the family Gelidaceae. And this agar is otherwise known as Japanese isinglass, or it is also known by the name vegetable gelatin. Then we'll move on to the chemical constants of agar. So agar contain two important polysaccharides, that is the agarose and agaropectin. And this agarose is responsible for the gel strength of agar, whereas the agaropectin is responsible for the viscosity of the agar. And the main uses of agar are, it is used as a very good gelling agent, then it is used as a laxative, emulsifying agent, then it is used in the preparation of suppositories, pessaries, etc. And the major substituents for agar include the Danish agar or Indian agar. This is a brief summary about the agar. Then we'll move on to the detailing of agar. So as I told you, the synonym is nothing but it is a vegetable gelatin or Japanese isinglass. And coming to the biological source, whenever we are telling the biological source, we can include the genus name, species name, family, and whether it is a dried form or the fresh form, or it is an extract, or it is a gum, etc. So we can say it is a dried aqueous extract which is obtained from by concentrating the decoction of the various genuses of Gelidium amansi, Gelidium elegans, which belongs to the Gelidiaceae family, and also from Pterocladia, that is again belongs to the Gelidiaceae family, and another species are available for the preparation of agar, that is a Gracilaria, which belongs to the Gracilariaceae family. And when we study about the morphology of the agar, they are yellowish gray or nearly white to colorless in nature, and it is odorless, and they are salty and mucilaginous in nature, and this mucilaginous taste is due to the presence of mucilage in agar, and they occur in different forms like strips, sheets, bands, flakes, coarse powder, fine powder, etc. And this shape depends upon the size of the various agar particles. And coming to the solubility, they are insoluble in cold water, but when we add cold water to it, it swells and it dissolves in hot water, and on cooling it forms a jelly. And the major chemical constituents include the agarose, which is a neutral galactose polymer free from the sulfate ion, and is responsible for the gel strength and mainly composed of galactose and anhydrogalactose units. Whereas if you see the agaropectin, it is responsible for the viscosity of agar, and it is a sulfonated polysaccharide in which the galactose or uronic acids, they are partially esterified with sulfuric acid. Now we'll move on to the identification test. That is, first we'll discuss about the solubility of the drug. As I told you, the solubility, it is soluble in boiling water and it forms a gel on cooling, whereas it is insoluble in cold water and organic solvents. But if you are adding the cold water to it, it will swell. And when a small little amount of agar particles is taken and it is mounted in the ruthenium thread, and if you examine it under the microscope, you can see the particles of agar will acquire a pink color. This is mainly due to the presence of mucilage in agar. And next confirmatory test is with iodine. So if you're taking a little amount of agar particles, and if you're adding N/50 iodine into it, you can see a crimson brown color. The next one that is on about the chemical test, which shows the presence of sulfate ions. So when we discussed about the chemical constituents of agar, we have seen that agaropectin is one of the important chemical constituent. It is responsible for the agar, and it is a sulfonated polysaccharide in which the galactose and uronic acids are partially esterified with sulfuric acid. So in the chemical test, if you are doing the hydrolysis of agar, that is, if you are taking a little amount of agar, if you are adding water, and if it is boiling for 30 minutes in a water bath, the hydrolysis occurs. That means in the agar, we have the agaropectin. So it undergoes hydrolysis and it forms galactose and uronic acid compounds. Along with that, the esterified sulfuric acid will undergo hydrolysis and form sulfate ions. So that is why when we are adding barium chloride into this hydrolyzed solution, it will give a white precipitate of barium sulfate. Then move on to the preparation of agar. So agar, as you know, it is a seaweed. It is collected from the sea, mainly in the month of May and October. With the help of the sea divers, or if we are inserting some long poles or bamboo sticks into the sea near the seashore to promote its growth on the bamboo. Then during the month of May and October, we are collecting the seaweed, that is the agar, with the help of the divers, or we can scrape it from the bamboos. Then it is dried in the seashore. So while drying in the seashore, there are chances of mixing it with the sand and the shells. So after drying, we are beaten and shaken to remove the sand and the shells. Then they are taken to the factories of high altitude where they are washed properly to remove the foreign particles as well as any salt which is present in the agar, and then they are dried properly by bleaching in the sun. Then they are boiled by acidulated water for about 5 to 6 hours, and then they are filtered while hot. Why? Because when we are filtering it while cool, in a cooling condition, that will get a gel form. So the care should be taken to filter it while hot itself. Then it is poured into the wooden troughs and is allowed to cool, where we will get the shape of the gels, and it is allowed to pass through the wire nettings to get the small particles of agar, and it is cut into different shapes like ribbons, flakes, coarse powder, etc. And finally they are dried at 35 degrees Celsius. And the next one, that is the gum acacia, or it is known as the gum arabic. So gum acacia is otherwise known as Indian gum. That is other name of gum acacia, and it is a gummy exudate which is obtained from the stem and branches of the Acacia arabica or Acacia senegal, and it mainly belongs to the family Leguminosae. And while making it is obtained by making the incisions, and while making the incision, the gum oozes out from the tree, and it occurs in the form of the round tears, which usually appears as a whitish or yellowish in nature, and they are usually odorless, and the taste is mucinous. And it is mainly, coming to the chemical constituents, it is mainly composed of two important chemical constituents. One is a compound, namely arabin, which is a complex mixture of calcium, magnesium and potassium salt of arabic acid. And it also shows the presence of two enzymes, that is the peroxidase enzymes and oxidase enzyme. And coming to the chemical test, this acacia contains the enzymes like oxidase and peroxidase. So to detect the presence of oxidase enzyme, first we have to take the aqueous solution of the drug, and to this we have to add few drops of hydrogen peroxide and a few drops of benzene in alcohol. You have to shake properly and allow it to stand. You can see a blue color developing on it, because this is due to the presence of oxidase enzyme. And next one, to the aqueous solution of the drug, if you are adding 1 ml of lead subacetate solution, you can see the formation of a white flocculent precipitate. Then if you are adding 1 gram of the powder, to the N by 50 iodine, usually an yellowish brown color is produced, which is a negative test, and by this test we can distinguish the acacia from the agar. And the major uses of agar includes a demulcent, emulsifying agent, suspending agent. Then it is used in the food as well as the other industries. Then adulterants includes the gum gatti, starch, tragacanth, dextrin, sterculia gum, etc. Thank you, students. Next class we will see the other plants which contain the carbohydrates.